Welcome back to Masterclass in Biology once again. We are discussing the basic concepts of photosynthesis in higher plants. And this is our final revision in Masterclass before the NEET entrance examination. And we are trying to understand all chapters in entirety so that we are better prepared for NEET entrance exam. As I always say, that flexibility is a very, very important tool that you should have in your armory if you want to become successful in life. This is one chapter which is very concept heavy. As you can see that in plant physiology, what I am trying to do is trying to tell you in brief the basic concepts, so that if these basic concepts are absolutely clear, the second part, which is learning of the facts, would be much easier. As I already told you that there are a lot of questions to practice, especially in the understanding NCERT section, all with audio solutions. So that would be a very useful exercise after you watch this video. If you think that you need more exposure to the questions, you can obviously listen to those answers for sure. Now again, continuing this, now this video would be on the second phase of the photosynthesis, what are called as light independent reaction. Obviously it is very important that you have watched the first video where I have discussed very important points. And if we are ready, if you have read the NCERT, then let us look at this particular phase of photosynthesis where the inorganic carbon is getting fixed into organic carbon, and this is where the synthesis part of photosynthesis actually takes place. Now, I will tell you something that we already know that if the plant has ATP and NADPH, then as long as these are available, it can carry on the fixation of the inorganic carbon into organic compounds, and this thing will be happening in a light independent phase. Now let us understand this particular part, what exactly is meant by light independent. Now light independent means, as I told you about Blackman's experiment, that this is a phase that is happening in the stroma of the chloroplast where some enzyme mediated reactions are leading to carbon fixation. Number one, this is something important to note. This is dependent on the enzyme, as I have already told you, and please remember that it does not require light directly. Now it requires the product of the light reaction, namely source of energy, which is ATP, and a reducing power, which is actually NADPH, a cousin of NAD, as I told you. So please remember two very important things I have told you: source of energy, which would be ATP, that would be a product of light reactions, as well as a reducing power. This time this is actually nicotinamide adenine dinucleotide phosphate, NADPH, rather than NADH. I hope this thing is absolutely clear. Many enzymes of the dark phase are also light activated. So that is what we are trying to say, that now we will not call it as a dark reaction. We are saying it as a carbon fixation, or we are saying it as light independent reactions. Now, majority of the plants that you will see are what are called as the C3 plants. So if you look at the common grain plants, say for example wheat, rice, they are C3 plant. What does it mean? So there is a cycle that is going on, and as you can understand, as soon as I say cycle, it is very similar to the Krebs cycle that we were talking about in respiration, in the fact that something will be entering into the cycle, there would be a product of the cycle, and something would be regenerated. So the same type of thing is going to happen in this cycle as well, and this cycle was actually deciphered by Melvin Kelvin, and in his honor it is actually called as a Kelvin cycle. And he also used the isotope tracing method to see the steps of the Kelvin cycle. Now we know where is the location of the Kelvin cycle. You know about a leaf anatomy, you know about the mesophyll cells. This is, I will skip it. You know that there are large number of mesophyll cells. Mesophyll cells have large number of chloroplast, and we know that what is happening inside the chloroplast. Now, Now, what we must understand is that this carbon fixation step would be that this inorganic carbon will be fixed into an organic compound. Now, this is the first step where we will say that this is the initial carbon fixation. Now, this carbon dioxide molecule has one carbon, and in the Kelvin cycle we will get a molecule as the end product, which is actually G3P. So this G3P molecule has 3 carbons. So what I am emphasizing here is that one by one 3 molecules of carbon dioxide will be getting incorporated. So to make one molecule of 3 carbon G3P, you will require 3 turns of the Kelvin cycle, and this is what we are discussing. So what I am showing you is actually 3 turns of the Kelvin cycle, and which will lead to a 3 carbon product. This is actually G3P, and 2 G3P molecules will make a 6 carbon product, which will be called as glucose. Certainly not a reversal of the reactions of the glycolysis, but you can get the idea that 2-3 carbon molecules can actually join to form what is called as glucose, and therefore there would be, to make glucose, there would be 6 turns of the Kelvin cycle. I hope this thing is now fundamentally absolutely clear in the mind of the students who are hearing us. So we say that 3 molecules of this carbon dioxide enter this cycle, the cycle that we are calling as the Kelvin cycle, and there is a molecule with 5 carbons. So please look at this. This is 5 carbon molecule, and it has phosphate and phosphate attached to it, and that is why it is called as a biphosphate, and this is actually RUBP. Now this thing is having 5 carbon. So we are looking at 5 carbons, obviously 3 molecules of 5 carbon atoms. So now you have total 15 carbon, and in 3 cycles 3 carbon dioxide are entering, and therefore it makes a total carbon number of 18. So this is 18. So what happens is that they join together. There is an intermediate form which is energetically very, very unstable, and then actually it cleaves itself, and it leads to formation of the first stable product of this cycle, which is actually a 3 carbon atom molecule. So there it contains 3 carbons, and this thing is called as 3-phosphoglycerate. So this is 3-phosphoglyceric acid. I hope this thing is now absolutely clear. This is the molecule. This molecule, because it has 3 carbon present in it, and it is the first stable product of this cycle, we are calling this cycle as the C3 cycle. And in the C3 plants, this is happening in the mesophyll cells. I hope this is absolutely clear. And this step you will call it as a carbon fixation. So this is the step you are calling as carbon fixation. I hope this thing is now absolutely clear. Now there would be reduction. Now the second phase would be reduction. What is going to happen is that first you will use the source of energy. Remember, you require 2 things from the light dependent reaction. One, the source of energy, and second that you require is actually reducing power. And I have told you why NADPH will be a very, very good reducing power, and obviously this would be 6 molecules of them. You have to account for the 18 carbon that you have started with. So this is 6 molecules. So you are using for phosphorylation the chemical energy that is stored in ATP. So you cleave ATP, and this generates ADP, and in organic phosphate you phosphorylate the 3-PGA, and 1,3-BPG is formed. So now just it looks like something happening in reverse when we think about the Krebs cycle. So this is 1,3-biphosphoglycerate. So where is the other phosphate coming from? It is coming from the hydrolysis of ATP. This is the source of energy. Now the reduction will happen. So for reduction you require reducing power. Obviously you will be using 6 ATP molecules, and 6 ADP will actually be formed. I hope this is absolutely clear. 1 for each molecule of the 3-PGA. This is, I think, now very, very simple. Now you have to reduce this thing. Now you have to reduce this thing. You will reduce this thing into G3P. So you are going to reduce this thing into G3P. Obviously, this also contains 3 carbon and 6 molecules. Again, we are keeping track of 18 carbons here. And now you require a reducing power. And that reducing power was NADPH, and that was actually the product of the light dependent reaction and is present in the stroma. So do you remember that where NADPH was actually formed? And therefore, this is where you actually use 6 molecules of NADPH, and this also actually this will actually regenerate NADP plus, so that the copper the thing actually continues. So this is where the reduction happens, and now you have 6 molecules of 3 carbon G3P. Still 18 carbon are intact. Now you pull out one. This is the end product of the Kelvin cycle. So what you are left with, if you pull out 1 out of the 6, now you are left with 5 G3P molecule, each having 3 carbon. So this is 15 carbons. Now we have to regenerate. From 5 molecules of 3 carbon which are having 18 carbons. I have to regenerate 3 molecules of 5 carbon, and you can see 5 into 3. 5 into 3 will be 15 and 3 into 5 will be 15. So that means with the help of 3 carbon containing 5 molecules, you are generating 5 carbon containing 3 molecules. This is the step, the third phase of the Kelvin cycle, the third part of the Kelvin cycle after carbon fixation reduction. This is where the regeneration actually takes place. So these are the 3 distinct parts of the Kelvin cycle, and obviously this will require more energy, and because what is resulting is actually 3 molecules of RUBP that are regenerated by Kelvin cycle. So you actually will have to use 3 molecules of ATP, and they will be hydrolyzed for this, and this is how it is regenerated, and the cycle is actually repeated. So this is for 3 turns of the cycle. With 3 turns, you get 1 G3P. Now you can calculate what happens if we are having 6 turns of the Kelvin cycle. This is how they are going to ask you the question. And what happens if we are thinking of only 1 turn of the Kelvin cycle? That would be very, very easy to understand. Now, to get a G3P molecule, now look at this. You are requiring around 1.5 times higher ATP number of molecules. So the ATP number of molecules, the number of ATP molecules I should say, that are required for the production of G3P, they are actually 9. So you are requiring 9 ATP, 6 here and 3 here. So this number is 9, but you are requiring only 6 NADPH molecules. I hope this thing is now absolutely clear. And since 2G3P will produce a glucose, so if we are looking at the glucose molecule, immediately you can see this number would be 18 ATP and 12 NADPH. And if you look at the non-cyclic flow of electron in the light dependent reactions, then you can understand that normally the number of ATP and NADPH should be equal in the non-cyclic flow. But for carbon fixation in the Kelvin cycle and the regeneration of RUBP were to continue the Kelvin cycle reactions, you require slightly more ATP. This is 1.5 times 18 per glucose molecule rather than 12 NADPH. So somewhere you will have to make this correction. This imbalance has to be corrected, and as we told you that this happens when there is a cyclic flow, and in the cyclic flow ATP is produced, NADPH is not produced, and oxygen is not evolved. And this is something where actually this thing can happen. You know about the stroma lamellae, and they will have only photosystem 1. They will have only photosystem 1. This is the place where this thing might be happening. This is an important question for your NEET entrance examination. So I hope this simple thing is now absolutely clear. Now, an important question would be about a very, very important enzyme. Now, this enzyme is called as Rubisco. So this enzyme is called as Rubisco. Now, this enzyme is probably the most common protein in the biosphere for obvious reason. You can understand this is an enzyme, this has to be a protein, almost all enzymes are protein, and we are saying that this probably is the most common protein in whole of the biosphere. Just understand the importance of this enzyme. Unfortunately, the problem is that yes, this enzyme actually has an carboxylase activity but also has oxygenase activity under certain conditions. And under certain conditions, it could actually add oxygen to the Calvin cycle. So rather than adding carbon dioxide to the Calvin cycle, where this inorganic carbon actually gets fixed into the organic carbon, what it can do actually sometimes is that it can actually add oxygen to the Calvin cycle with very bad consequences. So this is especially seen when the temperature is high. So when you are looking at arid conditions where the temperature are high and you are looking at some plants which are growing there the Rubisco has a problem. It has an oxygenase site as well. It can add, again I am saying, oxygen to the Calvin cycle, and thereby actually it can lead to production of carbon dioxide, and the fixed organic carbon can actually be oxidized and carbon dioxide may be actually released, and this will lead to a loss of photosynthesis in the plant. And this is going to happen when the temperature is high and is therefore is called as a photorespiration. So we have already seen the equation of respiration. In respiration, actually what happens is that the organic carbon is actually oxidized and carbon dioxide is produced, oxygen using oxygen. The same thing is happening because of Rubisco, and this particular phenomena is actually called as photorespiration and can be very, very bad for the plants, for the efficiency of the plants which are actually present in the regions where growing in the regions where the temperature sunlight is actually very, very high, and this thing is actually called as photorespiration. So this is a problem, especially a problem for the human beings, because we are dependent on the plant for so many products. I have already told you about wheat. I have already told about this wheat plant. You know, it is one of the staple grains consumed, and it is actually a C3 plant, and in them this Rubisco enzyme, as I say again and again, this Rubisco enzyme has an oxygenase activity as well. In normal circumstances, under normal temperature conditions, under normal light condition, what is going to happen is that the stomata will remain open. Now let me tell you a very important concept. So what is happening is that plants, they have some unusual problems. So they have to adapt to the terrestrial life. Like all terrestrial organisms, plant must also conserve water, and therefore you know about transpiration. So this transpiration is also occurring through stomata, and so does the gaseous exchange. So that means the plant cells get carbon dioxide and the oxygen is released into the atmosphere. This gas exchange actually is taking place through stomata. So is the transpiratory water loss. So evaporatory loss through transpiration is also occurring at stomata. So this is a trade-off. It may become a necessary evil. So if you want to keep the stomata open, or inevitably it will lead to transpiration. And what happens when the conditions become such that the transpiration becomes very, very large, and there will be huge water losses, and this can have very adverse effect on plant growth. It can lead to wilting. It can lead to so many problems, and ultimately it can actually affect the health of the plant very adversely. So what plant does is that it actually closes the stomata. So whenever such conditions are there, it closes the stomata. Now closes the stomata. Now what happens in the leaf spaces? So the carbon dioxide gets consumed and concentration of carbon dioxide dramatically falls. And because the photosynthesis is taking place, and you know that one of the byproducts of photosynthesis is actually oxygen, and oxygen gets accumulated. As long as the concentration is okay, carbon dioxide is more or even equal to oxygen surrounding the Rubisco will have more of the carboxylase action and oxygenase activity will be low. But what happens when the concentration of carbon dioxide gets low and oxygen accumulates? Suddenly you will see that it will be starting to add oxygen to the Kelvin cycle, leading to the loss of the fixed carbon in the respiration, and this thing is called as photorespiration. I hope this is absolutely clear. So again, understand, when these type of conditions are present, plants are likely to close their stomata, because at any case they will have to conserve water, and this will have some very bad consequences with respect to photosynthesis, because in the leaf spaces the concentration of carbon dioxide will have a dramatic fall and there would be an accumulation of oxygen, and this will encourage the oxygenation function of the Rubisco. Probably in the earlier life when the oxygen content was not very high, it didn't matter. This type of activity didn't matter. That is why probably it was not selected against. But now the oxygen is present in quite a big amount in the atmosphere, and therefore it becomes very, very significant in C3 plants that they can actually have a loss of the fixed carbon. The photosynthetic yield may actually decrease significantly. It can go up to probably more than 50% even, and this much amount of carbon dioxide, carbon fixed into the organic compound may be lost to a process which is called as photorespiration. Now the question is that why this photorespiration is actually occurring. So we say that it is not producing ATP. It doesn't produce any ATP, and actually it doesn't fix carbon. So no carbon fixation. Actually, on the other hand, what is happening? There is other way around. The fixed carbon is being lost to respiration, and this is the loss of photosynthate. Now, why exactly is it happening? And moreover, when the plants try to overcome this problem of photorespiration, we will see they will have to use ATP. So rather than producing ATP, it is using ATP in this process, and because respiration uses ATP, and it is leading to the loss of already fixed carbon. So it is not getting any advantage to the plant. So why is it happening? So most scientists actually agree that this is an evolutionary relic. As I told you about the Rubisco, it probably didn't matter the oxygenase activity in an environment where the concentration of free oxygen was not very high, but now it is becoming very, very big problem. So I hope you can understand this may be an evolutionary relic, and a few scientists also argue that it may also have a protective effect for the plant. So the debate is still going on, but definitely it is true that it definitely has to be an evolutionary relic as well, and we have to know more about the protective effect of photorespiration. If any, otherwise it is a waste process, it is a useless process. Now, how does the plant salvage this situation? So this is inevitable. If the temperature is going to go high, plant will definitely close stomata, and then you can understand the problem of photorespiration is inevitable in the C3 plants. So what can be the strategy? So the plants have two different strategies to combat this problem of photorespiration, and this is where one of the most important questions are asked. One of the most important question gets asked in your NEET entrance exam. And the first is that some plants carry out what is called as the C4 pathway. So this is C4 pathway. It is also called as Hatch-Slack pathway after the name of the scientist. This is something probably the most important question that is in the past years has been asked from photosynthesis. Yes, anything can be asked. Light reaction is equally important, but because they are asking few questions from photosynthesis, which is actually a very sore pain point for me, they should ask more from photosynthesis. You can see how many good concepts are actually there. They should ask one from light reaction, they should ask one from the light itself, and one from C3 and one for C4. So I am all for at least 3 or 4 questions should be from photosynthesis and another 2 or 3 from cell respiration. This is what I want. But I am not making the NEET entrance examination. I am merely preparing myself for the pattern that actually appears in the NEET entrance examination, and this is where most of the questions have been framed. So what happens in these plants is, say for example, the plant may be sorghum, so there are some maize. Please try to remember these plants. These plants are C4 plants. What does it mean? So what was the meaning of C3? So C3 means that the initial carbon fixation led to the first stable product which was having 3 carbons. So what would be C4? So C4, the initial carboxylation would be leading to the first stable product that will be actually a 4 carbon molecule, and this is oxaloacetate. And these type of plants will have what are called as C4 photosynthesis. This is the basic thing to remember. Now what I am saying is that in these plants, the leaves have a very special type of anatomy. So I am going very fast here. Please try to see. They have some very special cells. These cells are very near the veins of the leaves. These special cells, they are called as the bundle sheath cells. So very impervious, very thick walled. They are near the leaves, near the veins, and like a wreath. And there you can see these type of cells. They are called as bundle sheath cells, and obviously they have nearly placed mesophyll. So mesophyll cell cannot be very distant from the bundle sheath cell. This is something very important, and there may be there are actually plasmodesmatal connection between the two as well. So now we are looking at a dimorphic type of leaf. This type of anatomy is called as Cran's anatomy. And we are looking at two distinct types of cells. Obviously the normal one, which are called as mesophyll cells, and the second one which is called as the bundle sheath cells. So this is Cran's anatomy. There are different type of chloroplast. I have told you about the proximity to the vascular bundle. I have told you about the thick walled nature of the bundle sheath cells. They are very, very closely packed, and obviously they are near to the mesophyll cells as well. Now what happens in C4 plant is that the initial carbon fixation or carboxylation reaction occurs in the mesophyll cells, and the bundle sheath cells carry out the Calvin cycle. So here they are spatially separated. So that means the carbon will be fixed by twice, and the Calvin cycle carbon fixation will occur in the bundle sheath cell, not in the mesophyll cell. So mesophyll cells do not have Rubisco. So mesophyll cells do not have Rubisco. Instead, they have another very, very important enzyme. You must be knowing it. This is called as Pep Carboxylase, or what is called as pepcase. It has a very, very high affinity for carbon dioxide and has no affinity for oxygen. And this is the compound. This is the enzyme that actually carries out the initial carbon fixation or initial carboxylation reaction in the mesophyll cell, where the carbon dioxide joins what is called as PEP, and it leads to the formation of the first stable product, which is oxaloacetate, which has actually 4 carbon. So this is where this organic acid is formed, and let us say that this organic acid that is formed is oxaloacetic acid. It has 4 carbon, and now you know that what are the two molecules that have combined. So one is phosphoenol pyruvate, the second is carbon dioxide, and pepcase having a very, very high affinity, no oxygenase activity at all. Now that means this is where the organic acids, now these organic acids, say for example this oxaloacetic acid can be converted into another acid, a 4 carbon acid, which is actually called as malic acid. This is one of the most common things to happen, and this malic acid through the plasmodesmata gets transported into the bundle sheath cell. So in the bundle sheath cell, we know that the Calvin cycle is going to take place, and this is where the Rubisco is present. But what is reaching here? Not oxygen. Not oxygen. So oxygen is not reaching here. What exactly is happening here is that the Rubisco, in the vicinity of the Rubisco, you have a 4 carbon organic acid that gets decarboxylated, and it regenerates a 3 carbon pyruvate. It regenerates 3 carbon pyruvate. The carbon dioxide gets into the Calvin cycle and is fixed as normally in the C3 plant. So this is where the C3 cycle is actually happening in the bundle sheath cell. When this is getting decarboxylated, the carbon dioxide gets accumulated around the Rubisco, and it will have a carboxylase action, and therefore it will put the carbon dioxide into the Calvin cycle. This carbon dioxide is getting fixed, and what remains after removal of 1 carbon? It is actually 3 carbon. This is actually pyruvate. This is transported back into the mesophyll cell, and where with the help of ATP. where with the help of the ATP you regenerate phosphoenol pyruvate, and that continues the process. So I hope this is absolutely clear. Yes, you are using ATP molecule. That means it requires energy, but at least it is able to salvage the already fixed carbon and is able to reduce the respiratory loss. Actually, it is able to get the respiratory loss. Photorespiration doesn't happen in them for the simple reason the enzyme has been actually protected from the oxygen, and it doesn't carry out the oxygenase activity. So if you remember, there were 18 ATP molecules that were used in the fixation of the in C3 plant for 1 molecule of glucose. This would be more energetically expensive. It will require 12 more per glucose molecule, but it is worth. It is worth because it is salvaging the already fixed carbon. So this is the C4 strategy. In C4 strategy, as I told you, the C3 cycle does take place. The question would be that what is the initial acceptor, what is the first stable product, what is the enzyme, and where is the initial carboxylation reaction occurring, where actually the C3 cycle taking place. Now please remember, even in C4 plants, the C3 cycle is happening, but these two things, the initial carboxylation and the C3 cycles, are spatially separated. One is occurring in the mesophyll cells, the other is occurring in the bundle sheath cells. I hope this concept should be now absolutely clear to students who are watching this, and because this is just before NEET entrance examination, so I am not going into the various organelles and the reactions that are happening in the photorespiration. As I told you that in this MVQ series, we are trying to limit ourselves only to the NCERT, and we are not going outside NCERT, but it is again a very good concept. If you have time, you can watch the videos in our complete syllabus where we have discussed all the reactions that are happening in the photorespiratory pathway as well. But for now, I am skipping it. There is another strategy. This would be seen in succulents. So immediately you can understand that we are talking about the plants which are growing in arid region, very dry regions, desert-like conditions, xerophytic conditions, and you know that I am talking about mainly cacti. And you know what is succulent. The stem becomes swollen and it stores water. It becomes photosynthetic. The leaves are reduced to spine, thereby decreasing the surface area so that the transpiratory loss actually is minimized. And these plants, these are succulent plants. They do not have Kranz anatomy type of picture. So they do have only mesophyll cell. But what they do is that they close their stomata during day. So in the day they close the stomata to prevent the water loss. So this is the most important thing. Water is very, very important. So they close their stomata in the day but open the stomata at night. And at the night in the mesophyll cell, what is going to happen is that this carbon dioxide is getting fixed into the 4-carbon organic acid, and these mesophyll cells will store these 4-carbon organic acids in their vacuoles. And what happens during the day? In the day, if you remember, the light dependent reactions will continue, and the light dependent reaction, if they are happening, they will be producing ATP and NADPH. So now it has in its vacuole 4-carbon organic acid, and it has ATP and NADPH because the light reactions are going on in the day. The stomata is closed, doesn't matter, because this 4-carbon organic thing would be actually decarboxylated, and the Rubisco will be provided carbon dioxide and will carry out the carbon fixation by the C3 pathway. So here there is again 4-carbon organic acids that are formed during night. It gets decarboxylated in the day, and where the Rubisco doesn't get oxygen, it is actually getting more and more of carbon dioxide by the decarboxylation of this organic acid, and is able to carry out the carbon fixation by the C3 pathway. So this time it is happening in the same cell. Unlike the C4 strategy where they were spatially separated, they are separated in time. At night, the 4-carbon organic acids are being produced. They are decarboxylated in the day. That makes sense because in the day there would be light dependent reactions that will be happening. So you will have what is required for fixation of the carbon by Kelvin cycle, namely ATP and NADPH will be available, and the plants can carry out the C3 cycle. So what is very, very important to understand, whether it is C4 or it is C3, the plants actually actually all plants are carrying out the C3 cycle. This is something very, very important. There is a table in your NCERT. Now you try that table. There are some differences that are being asked, and please fill up that table. And I am very sure that one question in the NEET entrance exam this year may actually be between the differences between the C3 pathway and the C4 pathway. We have discussed conceptually all of them. What you need to do is right now open the NCERT and fill the columns in that particular exercise given in your NCERT, and that is very likely to give you +4 marks in your NEET entrance exam. Finally, there are some factors which affecting photosynthesis. We have already discussed in the beginning itself. Now there is one more point that I will tell you. The C3 plants, their photosynthesis rate will saturate at a much higher carbon dioxide level than what is present in the atmosphere right now. But the saturation actually in C4 plant is very near the carbon dioxide level that is already present in the atmosphere, in the present atmosphere. So what we are saying is that the concentration of carbon dioxide presently in the atmosphere is a limiting factor for C3 plant. So if you increase the carbon dioxide, you are likely to see what is called as CO2 fertilization effect, and it can actually positively impact the amount of the photosynthesis and the rate of photosynthesis. But there is another factor that needs to be taken into account. What happens to the opening and closure of the stomata, and is carbon dioxide a radiatively active gas? Can it contribute to the global warming? If the temperature increases, then what happens to the stomata? So things are very, very complex. But this is absolutely true that for C3 plant, the present levels of carbon dioxide in atmosphere can be limiting. Yes, if it is increasing, you are expected to see increase in the rate of photosynthesis in C3 plant, but C4 plant, their maximum is near the present concentration of carbon dioxide in the atmosphere. We already have talked about the light, the quality light, the quantity of light, you know about the wavelength, etc. You know about which are more energetic, and we have already told about the Blackman's law, the Blackman experiment, where we know how temperature is going to affect the plant as well. Please try to remember, plants have to keep in balance their requirement and the loss of water through the transpiration. This is a very important trade-off in the plants to be successful on the land in the terrestrial environment. And you know that angiosperms are very, very successful plants, and very, very great adaptations are seen in plant so that they are able to thrive actually in terrestrial environment. I hope you have understood the basic concepts in this chapter. Now you can practice all the questions. I am absolutely sure that you will find them very, very easy. Thank you for your time. Thank you so much.